Thanks so much, Mike, and
Thank
good
you, Mike.
morning, everyone. My name is Sharon Watson from the Office of Scientific Professional Development within the FDA Office of the Chief Scientist, and I'm pleased to welcome you back to Day 2 of the 2021 FDA Science Forum. The theme for this year is Science as the Foundation for Protecting and Promoting Public Health. Members of cross-agency science forum working groups have volunteered countless hours over the past year to organize this forum. Thus, I'd like to take a moment to give special thanks to the Science Forum Planning Committee and various work groups, all of our presenters, the CDRH Studio, FDA's web team, our AV team, Catherine Ng of Orsi, and Mike Kaczynski, who you just heard from, and the CIFSAN Staff College for all of their assistance for this platform. Today we'll have 4 concurrent sessions that present the amazing diversity of the scientific research at the FDA. Please refer to the Science Forum's website for the webcast link to your preferred sessions. The 2021 FDA Science Forum virtual poster sessions, as mentioned, are also exhibited on FDA Science Forum's website. The posters are available for download to all FDA Science Forum participants. In addition, the audience will have the opportunity to email their questions directly to the designated poster authors from May 26th through June 9th, so you'll have plenty of time to view those. As mentioned, this event will offer continuing education credit for viewing the various presentations, so please view the FDA Science Forum webpage regarding CE for your preferred sessions. Lastly, you will receive a survey to evaluate the FDA Science Forum in the next few days. Please take a few minutes to complete the survey as we value your feedback to improve FDA Science Forums. Again, thank you so much for joining us today. Without further ado, we will begin today's event in this room with concurrent session 5, Advancing Products Based on Novel Technologies, where Beverly Lynn will be our moderator for this session. Beverly, the floor is yours.
Good morning and welcome to Section 5. In this section, we will discuss the potential use, the limitations, and challenges of new technologies such as microphysiological systems, the microbiome, and the combination of both in regulatory science and advancing product development. Today, we will discuss in a series of talks the possibility of integration of these novel alternative models into potential regulatory environments that can be used to address unmet public health concerns, better models of human adverse response, as well as identifying and evaluating more reliable biomarkers for monitoring and interaction with the microbiome. It is my pleasure to introduce our keynote speaker to kick off this section this morning, Dr. Linda Griffin, Professor of Biological and Mechanical Engineering, Massachusetts Institute of Technology. After Dr. Griffin, we will hear from scientists representing all the centers of the FDA. Dr. Griffin.
Thank you so much for having me here today. I was asked to share with you some of the things we've been doing to overcome challenges in culturing super strict anaerobes that grow in the colon together chronically for days at a time with a healthy human mucosal barrier. And when we approach this problem, I've worked in the field of microphysiological systems since before it even started in the 1990s. We built models of liver and we built some other models. But the gut was a particularly challenging one for a number of reasons. First, what do we model? And functions of the gut depend intimately on the integrity of the mucosal barrier and its interactions with the immune system. So there's crosstalk not just between the microbes and the epithelia, but also the immune system and even the systemic circulation. So we started out thinking about particularly disease modeling, but these issues also arise in how drugs interact with the mucosal barriers because many facets of metabolism, transport, et cetera, are dependent on the proper functioning of the mucosal barrier. So we think a lot about first what to model. For example, in Crohn's disease, there's need to model parse the influence of genetics, the influence of particular immune alleles, and so on. So we would like systems that allow us to come in and parse the human condition in a controlled way. If we look further ahead, we're not there yet. There's certainly a need to bring in the neural system. The enteric nerves play a crucial role in functioning not just of the epithelium, but also the immune component. So these are the kinds of questions and challenges that framed our approach in thinking about this. And of course, there's a huge force because of the lack of fidelity of common animal models to reproduce the human condition, particularly when it comes to the gastrointestinal tract. We eat different things, we have different microbiomes, there's different kinds of immune defenses in the mucosal barrier, and there are many instances, and this is just one, of when something appeared to be very effective and exciting in a mouse model, and then taken into humans, it turns out not to work. So we're motivated by how can we start to build these systems in a way that companies and academic investigators can use them to look at focused questions of how the microbiome, mucosal barrier, particularly in the colon because it's the most challenging, how do they influence therapies? And so here's our wish list of what we want to build in a model. We want, and this is the wish list, we're not completely there yet. Elements of this are present in various labs throughout the world, but no one lab has combined every single. I think we've addressed the very, very crucial element of the co-culture of the microbiome with a version of a mucosal barrier, and we're heading toward this complete. So let me take you through. Of course, we need complete anaerobic conditions on the apical side, and anaerobic is sometimes a relative term. There are many microbes, even C. diff and others, B. theta, that are often classified as strict anaerobes, but in fact, they can grow when there's a teensy tiny bit of oxygen. Other microbes, as the one I'll talk about today, Faecalibacterium plasmidii, which we use as our test case, is completely unable to grow if there's even a tiny bit. So we use the most extreme to test our system. And we want to have continuous flow so that we can build up a homeostatic model. And so a primary cell culture, KCO2s, are certainly valuable in many situations, but using primary cells in this situation is highly desirable. We would ultimately like to have a crypt lumen structure, so the normal architecture of the gut is the stem cells reside here and cells move up and then are sloughed off into the colon lumen, and we would like to get that architecture because the stem cells are driving the differentiated cells, and then this whole process will continue over time, and this is maintained by gradients of growth factors and metabolites. Ultimately, we want communication with stromal cells, immune cells, a vasculature, and lymph drainage. So this is the wish list, and I think we can accomplish this within a couple of years, actually having models that incorporate all of these. However, we're not all the way there yet in putting all the elements together. So I want to share with you some of what we have been doing, or where we are. So two huge areas of technical challenge are first the platform to meet those requirements of the total anaerobic flow on the apical side and aerobic conditions to nourish the mucosal barrier on the basal side. That's actually really hard, especially if you want a continuous flow in both places so you can replenish the nutrients and have the spent cells and tissue washed out. We also have a lot of other features such as being biocompatible, being able to sample, etc. Then there's the tissue engineering side and biological function. How can we make a device that accommodates the 3D tissue, and how do we make the 3D tissue that incorporates all of those elements that we want? So these are the things I'll briefly discuss now. So when we started this project, there were a number, and since then, there were a number of gut-on-chip devices that had been published in the literature, and we can go down and look at them and compare with respect to the materials they're made of, for example. We want materials that are compatible with using lipophilic compounds such as steroid hormones, for example, and lipophilic drugs. So having materials that don't absorb those are important. How the cells are seeded and matured, how long you can do the co-culturing, whether or not there's flow, what kinds of cells are able to be maintained, the kinds of species, and whether or not there's direct contact between the bacteria and host, because there can be concerns about overgrowth of bacteria. So these are all the kinds of questions that we set out to try to combine in one device. So a major decision we made early on was to focus on using transwell culture. This is a very common mode of culture for barrier epithelium of all kinds, and especially for the gut. We leveraged the incredible advance that Hans Klevers brought to the field in making organoids, and then others in the field, who I mentioned, some of them here, have done tremendous work to show how you can build monolayers of mucosal epithelia from the colon and other parts of the GI system on a transwell. So you can take organoids and dissociate them and seed them onto a transwell, form a monolayer as shown here. So you see the monolayer here, the basolateral surface. This is from a publication by Nick Zakos. And then in many instances, if you want, you can add immune cells to the basal side of the membrane, and they crosstalk with the epithelia. These kinds of transwell systems are extremely well characterized in many labs, and investigators like using them. And so building a device that lets investigators take a familiar culture format that they trust and put it into a device that now allows them to culture with strict anaerobes was the direction we decided to take, with feedback from the community of users. And so the features that are the key features of the system, which was really built by two wonderful postdocs, Yuzhao Huang and Bob Zhang, is here. And so let me take you through it a little bit. So there's a transwell with a monolayer of the mucosal epithelium shown here, and completely anaerobic flow enters and flows across the top of that epithelial barrier. We can introduce microbes into this apical side after the epithelial barrier and flow have been established. We could also introduce pathogens at a later time if we like. And so one feature of this, this is actually not drawn entirely to scale, because this gap in our device is actually 3 millimeters. And 3 millimeters is gigantic compared to standard microfluidic devices, which typically would have a gap of around 0.1 to 0.3 millimeters. So it's about an order of magnitude larger than most microfluidic devices that have been used. And why did we do this? We did this because it allows us to keep that mucus layer that's on top of very, very intact, and allows microbes to settle in and populate the mucus layer while we flow fresh nutrients across the top. So we bring nutrients in constantly all the time, completely anaerobic, flow it across the top so the microbes have a continuous replenishment of their food supply, as well as we can then wash microbes out and build a homeostasis. At the same time, there is circulation on the basolateral compartment to provide oxygenation and fresh media to the mucosal epithelial cells. We can also, if we want, add immune cells on the basal side of this. So this is the first implementation, a standard monolayer, which does not last more than about 4 days due to differentiation processes that are natural. We're in the process of building a 3D culture, but a standard monolayer is very valuable for studies at the present time. So let me show you some published work on how well, first of all, let me just remind you that the approach that we took also leveraged micromachining technology that we had developed for a liver application and commercialized. That's actually a device that has been, is in-house at the FDA, and the FDA has published on this liver device. And an advantage of this approach is that these hard plastics don't absorb lipophilic compounds. So if you're trying to do pharmacokinetics and look at fate of the drugs, then you have an advantage in not having them absorb to your device. And PDMS allows them to absorb. Another feature of this device is that the pump system was adapted again from technology used for the liver chip. It's a tiny, tiny microfluidic pump that allows us to I'm just checking to make sure my phone is still on. It's a tiny microfluidic pump that allows us to have tiny, tiny little dead volumes so that you can squish the pumps and contain everything on one device versus having a lot of pumps wandering around and taking up space in the incubator and absorbing compounds. So that's a key feature that allows us to combine an anaerobic flow with an aerobic flow. Now, so this is what the device looks like in practice. I've shown you the schematic here, and there are 6 transwells together in a device that you set up and put in the incubator. This is a photo of it. When you maintain a colon monolayer in the device, it looks very healthy. You can see stain here. These are just immunostains. MUC2, these are making mucus. So that it's a healthy colon monolayer. And the features of the monolayer are such that we find a little bit of faster differentiation and repression of proliferation when we put cells from static culture into the GUNI device. So there's an upregulation of differentiation markers and downregulation of proliferation markers. This may be due to the fact that we have flow and a greater availability of nutrients for the cells in the device. Now that we've established that we have a healthy colon monolayer, we want to establish that we can grow a super strict anaerobe continuously with this monolayer and have some kind of biological function that seems similar to that in vivo in humans. And so if we look across the types of microbes that are present in the microbiome in the colon in humans, several of the very strict anaerobes, very strict is F. Prausnitzii, E. reptile, and B. theta is a little bit less strict, are very dominant in the human microbiome, as you can see here. So they're highly present, and many of these have been implicated in disease processes, particularly deficiencies of some of them have been implicated in inflammatory bowel diseases. So many labs have reported, for example, on F. Prausnitzii being implicated in ulcerative colitis and in other types of inflammatory bowel diseases. So across many different labs, this one seemed to be a particular microbe that, and one of the reasons it's implicated is that it is one of the major producers of short-chain fatty acids, particularly butyrate. So it ferments substrates that are present in the colon and produces butyrate, which can then act not only on the gut epithelia, but be transported across the epithelial barrier and interact with the systemic circulation and organs throughout the body. So we took F. Prausnitzii, and the first thing we wanted to show is that we could maintain a barrier function and even grow the bacteria and get them to be homeostatic over time. And so this is just showing the number of microbes over time, and this is F. Prausnitzii. So within 24 hours, we have what looks like a homeostatic number, and this is comparing to the CFU in the colon. And so then establishing that the bacteria are there by measuring them, we also then did some imaging to see where the bacteria are relative to the colon mucosal barrier. And so this is just a dramatic image of the colon mucosal barrier. The bottom of this would be the transwell. So this is after 3 days of co-culture, and you can see a dark layer here, which is very tight. In the colon, you have very tightly cross-linked mucus right at the surface of the epithelium, and then further out, there's more loosely cross-linked mucus where the microbiome takes up residence. And so this cloud of DNA here, so we've stained blue for DNA, this cloud here represents the bacteria growing in this lightly cross-linked mucus layer. And again, we don't have a thin gap, we have a huge tall gap between the surface of the epithelium and the top of our device, so the fluid can flow through gently and allow the mucus to sit there and host the bacteria in it. Now, importantly, and this is a little bit busy slide, so the important thing here is really looking at we're comparing static culture, the device without any bacteria, the device with bacteria, and then the culture medium that's fed across the apical surface to the bacteria. And we can see here, so we're looking at 3 short-chain fatty acids that are commonly examined, acetate, propionate, and butyrate. And butyrate is typically the one implicated in immune functions and inflammation. And we can see that only in the case of the presence of the F. prausnitzii bacterium, which is known to ferment butyrate, that we produce copious amounts of butyrate on the apical side. But important on the apical side, sorry, I'm getting used to using this coordinate. So the butyrate is here, acetate, propionate, butyrate. But in the basolateral medium, so this is on the other side of the epithelium, away from the microbes, in fact, we see copious amounts of butyrate when we have the fecal bacteria, fecal prausnitzii, Faecalibacterium prausnitzii present. So the butyrate is not only being produced by these bacteria that are alive, healthy, well, in their anaerobic microenvironment, they are fermenting their substrates and allowing the epithelial cells to transport them across the barrier, as they should, into the basolateral compartment. In humans, these butyrate would then act on immune cells. It would be taken up into the systemic into the portal circulation, go to the liver, and impact the function of the liver. So this is moving toward a natural physiological state for this monolayer. There's also impacts of the Faecalibacterium prausnitzii on immune modulators such as TLR3 and 4 and others. And this is still an emerging picture, and keep in mind we don't have immune cells present in this system, but there's clearly regulation of these TLRs, toll-like receptors that interact with microbes in the presence of the FP. We can also now go in and establish, and this is a manuscript in preparation, but I thought worth sharing, we can establish some elements of the immune system, and here we focus on elements of the innate immune system, monocyte-derived macrophages and dendritic cells, which can be attached to the basal side of the membrane so that they interact with the epithelia and can serve as sort of sentinels for things that the microbes may be doing to the epithelia. And so we can also include in the circulation CD4 positive T cells, for example, to look further at, for short periods of time, 2 to 3 days, to look further at interactions with the immune system. So again, even in the presence of these immune cells, if we look, there's no difference between the presence and absence of immune cells on the accumulation of a homeostatic number of bacteria density. It still looks the same as it did when the immune cells were not present, so there's nothing appearing to affect the bacteria, and the integrity of the monolayer is still quite robust. However, what we find is that there's a dramatic change in the baseline cytokine, chemokine profiles produced, particularly, of course, in the basolateral media, which you would expect because that's where the immune cells are. So we see a huge, mostly an increase in various immunomodulators. And then also, though, in the apical medium affecting the epithelial cells, there's an increase in things like IL-17 and IL-9 that we hadn't detected before in the absence of the immune cells. So I covered the platform. Let me finish up by describing some of the challenges in tissue engineering and then how we're trying to pull tissue engineering and platform advances together for a next generation that would be valuable in drug development. So again, we've focused on transwells so far. We leverage very strongly this organoid technology developed by Hans Clevers. And what he recognized is that the stem cell niche in the intestine maintains the homeostasis of the tissue. The stem cells reside in the crypt and they divide and the cells move upward and go into the lumen. And if you're in the small intestine, you go to the villi, and if you're in the colon, you go to the lumen. And these are maintained by growth factors produced by stromal cells here in the bottom of the crypt, so Wntz and other things. And so there's an intimate crosstalk. In maintenance of the tissue, there's also butyrate suppresses proliferation. So butyrate is going to make these cells more differentiated, but it doesn't penetrate down here. So there's a complex web of signaling that maintains this. A key advance in the entire field was the description by Hans Clevers and his postdoc Sato of how to grow these epithelial organoids in culture by taking advantage of driving stem cell expansion, putting cells in matrigel, basement membrane-like matrix that's extracted from tumor tissues. And so now the approaches for doing this have been described for mouse and human and iPS. There's a whole cocktail of growth factors used to expand the organoids in a stem cell state, and then you change the growth factors to allow them to start to differentiate if you're just growing the epithelia. So you grow just the epithelia, making up for the stromal cell production of growth factors by simply adding the growth factors to the medium. So labs all over the world use these approaches to expand patient-derived biopsies into tissue banks and do experiments. So now if we go back to our model, the thing that we want is, can we move toward growing a 3D structure that mimics features of this, including co-culture with the stromal cells and perhaps immune cells and so on. So to do this, we have over the past decade or so leveraged a common approach in tissue engineering based on polyethylene glycol branched polymers, 8 arms, as shown here on the left, that can be modified with small peptides that interact with cells or that bind matrix, a peptide cross-linker. These two reagents can be, and they're completely defined, so there's nothing, we know everything. We made it all in the lab. You combine these with cells and do a pH change that's very gentle, and you can make a beautiful 3D hydrogel like you see here that encapsulates cells in a very healthy way. And so we did a lot of engineering of this system. The basic system is there, but how you combine these reagents together to make certain mechanical properties, certain permeability properties, which peptides do you use, which integrin receptors do you target. These are all things that take a lot of engineering design, and I'm showing you what we came up with. We have what I call a one-size-fits-all matrix where there are peptides attached to the polymer, and these peptides bind to integrins, but there are also peptides that bind to some extracellular matrix like fibronectin or laminin or collagen 4 that are produced by cells, so that when the cell makes their own matrix, the synthetic gel now captures it and helps the cell assemble it so that it can remodel the environment so that it is the natural environment the cell would have in vivo. It is a very versatile system because it can keep both epithelial cells, which want laminin and collagen 4 in their environment, they produce it, and stromal cells, which have other types of collagens and fibronectin. It can keep both cell types happy, and also immune cells as well. So a wonderful postdoc in the lab, Victor Hernandez-Gordia, was able to do a semi-empirical screen of a parameter space of matrix properties, biophysical properties, biochemical properties, and he identified, and this is showing organoids from the intestine growing in a standard droplet format. Most organoid cultures done in just like 3 microliter drops on the bottom of the dish. And these are organoids that are grown in a completely synthetic hydrogel. They were grown from individual cells to form these stem cell, these were grown under the so-called stem cell expansion conditions. And what's essential is this peptide, the Gophoger, which we know interacts at least with alpha-2 beta-1 integrin, but probably others. And this is an essential peptide interaction because if we mutate one of the amino acids that is believed to be involved in binding with the integrin to a different one, just that one mutation to abolish binding, we lose the activity of the gel. So this is some evidence that it's working that way. So when we set this up, we screened it with a huge number of different donors, including mouse. Mouse is very easy to grow compared to human. And again, did a semi-empirical screen to arrive at a formulation that supports the emergence of Panna cells. They're characterized by this lysozyme, and they support stem cells. So we haven't definitively tested for stem cells. Those tests are rather hard, but this green indicates this cell next to the Panna cell is proliferating. So there are many features of these that suggest that we do have the normal kind of organoids that you would get in Natrigel. We're right now, this is work not published but ongoing by a student in the lab, taking these into a morphogenesis situation so that you could, in a somewhat straightforward protocol, allow cells to undergo a transition, if they're at the surface of the gel, into a crypt lumen structure. And this is very, very early days, but we're leveraging principles of morphogenesis, and again, biomechanics play some role in this to try to get these structures. And it turns out there's very important features that the stromal cells can play in helping this morphogenesis process. And this is very much a work in progress, but we're moving quickly. We collaborate with David Bro, and Victor Hernandez continues to work with us on this. So this is, we're moving toward allowing us to generate on a transwell in situ a true 3D structure containing these other cell types in the 3D structure. Now I want to finish up by just mentioning that a transwell is useful for now, and we are commercializing this device. Since I made the slides even, the company that has produces the liver chip is now going to start producing this device so that others in the lab can use it. We're in the process also of building an industry consortium so people can start to do their own experiments and test how microbes may be interacting with drugs or the microbes may be influencing biology themselves. But where we're headed is to combine the mucosal barrier with wonderful technology from my colleague Roger Kam, who pioneered approaches to build microvascular networks in microfluidic devices. So what's shown in the movie is this is a microvascular bed built in a gel in between 2 microfluidic channels, and the green were immune cells that are moving through the channels. We have recently translated all of our microfluidic technologies into a single platform that we're commercializing for use in this application so that we could ultimately marry the Gumi device I showed you with simple devices on chips. So I'll end there and thank my wonderful, wonderful collaborators on this project, primarily Dave Trumper, who's in the mechanical engineering department, Mechatronics, who led development of these, and Rebecca Carrier, a wonderful collaborator from Northeastern, whose lab focuses on nutrition and drug uptake in the gut. So I'll stop there.
Thank you, Dr. Griffin, for the exciting and inspiring presentation, not only addressing the challenges of these systems, but ending with the development of a platform. Our next guest will be Dr. Daniel Tedeschi from the Center for Veterinary Medicine. He will be further talking about advancing regulatory science through organs on a chip. Daniel.
Thanks, Beverly, for the introduction. Linda, that was a great talk. And for the next 10 minutes, I will give you a high-level overview of why we as a center are interested on the organ-on-a-chip technology and its potential to advance regulatory science in support of our center's mission. The effect of drug residues in animal-derived foods on the human intestinal microbiota is an important human food safety endpoint of concern that needs to be addressed during pre-approval evaluation of drug products intended for use in food-producing animals. This concern has been regularly addressed using in vitro and in vivo test systems. In the next couple of slides, I will discuss the limitation of existing test systems and what organ-on-a-chip potentially offer as an alternative to existing methods. Insight into the microbiome and its interaction with human hosts and their chemical environments can be obtained by using diverse non-human models. Non-human models can provide insight into the molecular pathways, physiological process, and microbial response to chemical stimuli. One example is animal models. Animal models are widely used to investigate the human microbiome for several reasons. It's much easier to manipulate animal models than human subjects experimentally. Animal models allow the careful control of experimental variables and reproducibility that's often important in human studies. There are, however, important caveats when we use animal models, including the salient difference in anatomy, physiology, diet, and microbiome difference between human and animal model. The other non-human model used to study microbiome-drug interaction is an in vitro system. The current in vitro system are either batch culture, with or without host interaction, or continuous culture without or with host interaction. The top two panels shows the most common system lacking living intestinal epithelium that have been used to study the gut microbiota. The bottom panels display the four current systems that have been developed to closely simulate human gut microbiota interaction. Recent advances in tissue engineering, microfabrication, and stem cell biology have enabled the development of sophisticated systems like organ-on-a-chip model that can recapitulate the key microenvironmental characteristics of the human or animal organs and mimic their primary function. I just want to give you a very brief definition of organ-on-a-chip. Organ-on-a-chip is a microfluidic cell culture system that recapitulates the in vivo tissue and organ level structure, function, physiology, and pathology in vitro. It could benefit humans and animal health, and it has a potential to lead to a paradigm shift in drug development, toxicological screening, personalized medicine, disease modeling. In short, it enables the creation of more in vivo-like in vitro models, and it has a potential to reduce and refine the use of animals in research. Microphysiological systems have been developed to model a broad spectrum of organs from heart, kidney, liver, to lung, gut, and many others to understand human organ development, disease pathology, and facilitate drug discovery. Broadly, we can divide this approach into two. The first being single organ platform, which integrates one specific type of tissue or organ and provide relevant microenvironmental context. For example, this figure shows intestinal microenvironment. It includes the 3D architecture of the intestine, the mechanical cues, the peristalsis movement and the flow stress, and the biochemical cues, the microbiome and the immune cells. Depending on the cues included, it can be a relatively simple cell or organoid-based intestine-on-a-chip model with intestinal cells cultured in a 2D monolayer, or it could be a more complex one with the 3D architecture of the intestinal cells co-cultured with multiple cell types, including the gut microbiome. The second approach, which is a more complex one, is a multi-organ on a chip system. This involves integrating multiple associated organ chips in a single platform, which allows the study of multiple organ function in a systematic approach. As I indicated at the beginning of my talk, FDA considers the effect of drug residues in animal-derived food on disruption of the bacterial colonization barrier and increase in resistance bacteria population in its pre-approval evaluation of drug products intended for use in food-producing animals. In light of this, we are evaluating emulate intestine on a chip system as an alternative in vitro model for co-culturing of human intestinal and microbial cells, allowing targeted perturbation experiment, specifically focusing two endpoints, which are disruption of colonization barrier and development of antimicrobial resistance. We are also evaluating the ease of use of the system, the variability, the reproducibility, and limitation. We are also looking at the duration. What I mean by duration is the number of days that the system maintains the cellular structure and function and mimics the human intestinal environment. We hope to also develop performance standards for validating the model. And finally, the validated model could definitely help pharmaceutical drug sponsors to provide data on the potential effects of animal antimicrobial drug residues on intestinal microbiota. In terms of our approach, we are collaborating with Emulate and Johns Hopkins, and we are coupling the power of microphysiological system and the next generation sequencing technologies. So we are using Emulate intestine on a chip system. The system has two channels separated by a porous membrane coated with extracellular matrix, with endothelial cells growing in the lower channel and epithelial or colonoid, in our case, growing in the upper channel in the presence of physiologically relevant luminal flow and peristalsis-like movement. In addition, we will be culturing complex human microbiota in direct contact with epithelial cells and create an oxygen gradient to mimic the intestinal microenvironment. As Linda indicated, this is going to be a very challenging task in terms of maintaining the oxygen gradient that's necessary for obligate anaerobes. However, Ingvar and his colleagues at the Wyss Institute at Harvard recently demonstrated anaerobic intestine on a chip model that can sustain over 200 unique operational taxonomic units from 11 different genera, including obligate anaerobe bacteria. They have shown that the chip stably maintained a microbial diversity like the richness observed in the human intestine. And we know that organoid culture have limitation in maintaining the cellular structure and function for extended time and sustaining the co-cultured microorganism for prolonged time. To make sure the system is performing as expected, we will be monitoring the morphology, differentiation status, and membrane integrity using microscopic evaluation, immunoassay, and biomarker analysis. We will be using metagenomics and metatranscriptomics approach to elucidate the microbiome host interaction as well as the microbiome host drug interactions. In closing, we hope that our research efforts help validate intestine on a chip model as an alternative in vitro model to support evaluation of new animal drug products. It could inform and direct pre-approval steps within and beyond FDA. To acknowledge Kathleen Wish, who's my team member, and my collaborators, Sylvia Pinero and Jeff Gilbert from Office of New Animal Drug Evaluation, who have expertise in regulatory science, Jean and Stella, who played a key role on the study design, Odile from CEDAR, and I would like also to acknowledge my collaborator from Emulate, Jordan, Ben, and Graciela, who are experts on Emulate flagship organ chip model for a variety of differential applications. And finally, I would like also to acknowledge Mark Danwitz and Nicholas Zakos, leading experts on the use of human enteroids to understand intestinal physiology and pathophysiology for their advice and providing the colonoid for the project. Thank you.
Thank you, Daniel, for that exciting talk and discussing the importance of these systems in advancing regulatory science. Our next speaker is Dr. Sangeeta Hare from NCTR. She'll be discussing the importance of the microbiome as an additional criteria for safety assessment.
Thank you, Dr. Beverly Lynn Cook. I would like to thank the organizer for providing me this opportunity to discuss my research at the FDA Science Forum. In next 10 minutes, I will be providing you the research outcome of my studies and would like to make you think about whether such approaches are useful for making the decision tree for GI safety assessment. So the goal of our study is to define the microbiome if it is adversely impacted by the xenobiotic exposure. And for this, we use the bottom-up approaches using the in vivo model. And second, we would like to develop the translational approaches using the top-down approaches using various in vitro systems and ex vivo systems. So the research tools that are used in my lab are to study the xenobiotic, microbiome, and host interaction. So for the xenobiotic microbe interaction, we start with simple experiments like MIC determination using the in vitro microbial culture, then move on to the batch culture, then ex vivo system, and the animal model. Similarly, we also use the xenobiotic host interaction using the in vitro culture system, either epithelial cells or monocytes, macrophages, and other cell types, 3D culture model, then human intestinal tissue explant, and animal models. So why an additional criteria for the safety assessment is needed? Because it is not sufficient to say that interaction of xenobiotic or drug may cause diarrhea. We need to know who is there and if they are metabolically active, and also what they do there. And for this, we use genomics, metagenomics, and functional genomics approaches using the holobiome assessment. So in our lab, we are using several xenobiotics and use this test system. So today, I will be mainly talking about the arsenic, how this can be used as a model xenobiotic to assess the xenobiotic microbe-host interaction. So arsenic is a ubiquitous xenobiotic, and the regulatory agency has defined the levels of this into the common food as well as into the dietary supplement and also in the infant rice cereal. The most interesting thing is that arsenic, when it goes into the gastrointestinal tract, it interacts with the gut microbiota and epithelial cells, and it can become more toxic by converting into the various toxic species. And this also causes concern for the gestational exposure and the intravenous exposure. So to study this toxicity, we have collaborated with Dr. Dawn Dorsey at the Division of Biochemical Toxicology. And today I will be defining the outcome of the two experiments. First is the adult exposure, in which mice were exposed with 3 different concentrations of arsenic, and the neonatal exposure. And the samples were collected just after 24 hours or the 48 hours after the exposure. So here in the result, you can see that the control is defined at very right in each of the graph. And here is the aerobic culture, anaerobic culture, and specific bacterial culture. And you can see that within 24 and 48 hours, there is decrease into the live recovery of the live bacteria. Similarly, then we look for the 16S sequencing, and here are various groups with the low dose, medium dose, high dose, and you can see the discriminant analysis showed a separation for different treatment groups. Then we went ahead and did the sequencing, and results are shown in the heat map. At the very left is the control, and you can see that all different experimental group has higher abundance or the lower abundance of the some bacterial species that are shown by the arrow. Same thing, we looked also into the developmental stage, and what we see here at PND21, there is a distinct population during the exposure with the arsenic, and it is also very different from the adult control. So next, we wanted to see what is the immune status, whether it is affected, and here is the snapshot, and we see that the high level of pro-inflammatory and anti-inflammatory cytokine secretion was affected. And we look for the functional analysis for the microbiome. Here is the snapshot of those results in adults at the top and postnatal at the bottom. So the bacterial species those are affected are mainly the bacterial species those are involved into the detoxification of the arsenic. And the lower abundance of the bacteria was those were involved into the butyrate production. And butyrate has a very major role into the gastrointestinal homeostasis. So thus, we wanted to see what could be the long-term effect of the arsenic exposure in relation to the GI toxicity. For this, we went ahead and did the in vitro culture using the intestinal epithelial cells. And here at different passages, we were looking for the expression of the genes. And here at the 14 passages, and these experiments are, by the way, still going on, the genes responsible for the progression towards the invasion and metastasis in gastric cancer were highly upregulated. So then we also wanted to see if the intravenous exposure is some different or the same way. So for this, mice were injected intravenously with the arsenic, and samples were collected 1, 4, 24, and 48 hours exposure. And the result here, when we compare the oral exposure to the intravenous exposure, were quite different. During the oral exposure, we found the bacterial species that were involved into the pathogenic bacterial invasion and were producing the immune responses, whereas when the intravenous exposure was done, there was disruption of the first protective layer and the epithelial layer disruption was seen. So next, we wanted to see if we can establish experimental model that can also be mimicked into the in vitro system. But before that, I would like to give you the results for how the oral exposure we see significant amount of signaling and inflammatory responses and no responses for the cell-cell junction, whereas IV exposure resulted in the downregulation of the cell-cell junction, whereas no major changes into the immune response related gene except after the 1 hour of incubation. And these were also correlated with the speciation of the arsenic that occurs to the oral gut microbiota. And we were able to see the several arsenic species, that is conversion of inorganic arsenic into the more toxic trivalent and tetravalent into the oral exposure, whereas in intravenous exposure, we were not able to see such species. So now we wanted to see if the in vitro system can be developed that can mimic the oral or intravenous exposure. So here the cells were cultured onto the transwell. Apical exposure mimicked the oral exposure and basal mimicked the intravenous exposure. And what we were able to see that there was indeed actually the changes into the TER when the exposure was done from the from the basal side. And same results were found for the expression of the genes that are involved into the cell-cell junctions. So next, our approach is to go for the translational research from the experience that we got from the lab animal model of how we can correlate it to the humans. So for this, we collaborated with Collaborative Human Tissue Network, and we obtained the biopsy punches or the tissue explant from them and culture it into 24-well transwell plate and do the exposure with the xenobiotics, and these experiments are ongoing. So with this, I would like to present you the decision tree for the evaluation. So here we can say that we start with the MIC determination, and then once if MIC is known, then it can further proceed for the animal study, and we can look for the abundance, we can look the metatranscriptomics, and we can do the metabolic profile. And if we find certain genes that are specific or bacteria specific, we can further investigate those. And similarly, we can do the host microbiome interaction also. So this contributes to the FDA strategic plan for advancing regulatory science. And we can say that this decision tree can form the microbial dysbiosis and intestinal toxicity be added as the endpoint for the hazard or preclinical safety assessment. And with this, I would like to thank my research team, and I would like to dedicate this presentation to Dr. Carl Seniglia, who passed away a month before. Thank you so much.
Thank you, Sagira. And because of time, we're going to press on to our next speaker, Dr. Zhuo Li from CEDAR, who will further discuss the microbiome. Dr. Li. You don't.
Oh, sorry, can you hear me? Can you hear me now?
Now.
Oh, sorry. Yeah, I was on mute. Thanks, everyone. My name is Zhuo Li from the Division of Applied Regulatory Science, Office of Clinical Pharmacology, CEDAR, FDA. Today, I would like to talk about one project that we have been doing, studying the emergence of nosocomial opportunistic pathogens in the gut microbiome after antibiotic treatment, as reviewed by a mouse model metagenome analysis. First, some standard FDA disclaimer. So, what are nosocomial infections? Another name for it is healthcare-associated infections, or HAI. These are infections associated with stays in hospitals, rehab centers, and nursing homes, etc. And many of these infections are caused by opportunistic pathogens with different types of antibiotic resistance. Shown here are three famous or notorious nosocomial pathogens: MRSA, or methicillin-resistant S. aureus, A. baumannii, and C. difficile. I'll talk about them more in details later. Nosocomial infections have huge impact. If you are admitted to a hospital, you have a 5% chance of contracting an HAI. Every year, 1.7 million people got infected through HAI, and 98,000 people were killed by nosocomial pathogens. So, why this? When we study the emergence of these nosocomial infections, where do these pathogens come around? There is a speculation that oral antibiotic therapies in the healthcare settings are linked to a nosocomial infection. My apology, but there's a typo on the screen. It should be antibiotic therapies, not antibody therapies. This is because it's well known that antimicrobial agents can kill others but allow antibiotic-resistant bacteria to grow in the gut. So it sounds like a plausible mechanisms to induce the emergence of such pathogens. However, if you ask, do you have any evidence of emergence of these pathogens as a direct consequence of using specific antibiotics? As far as we know, there are some evidence for C. difficile directly induced by clindamycin, but that's it. For all other pathogens, the in vivo evidence to link them to specific antibiotics is lacking. This is because to prove this direct link, you need to exclude many other confounding factors like diet, or baseline bacteria, other medications, etc. This is hard to do in a clinical setting. However, it is possible to control all these confounding factors in animal study, so we started using animal models. We first inoculated mice with uropathogenic E. coli to induce urinary tract infection, or UTI. And then we treated the mice with 3 different common UTI antibiotics: ciprofloxacin, fosfomycin, or ampicillin. And then we sequenced all genetic materials in fecal samples from these mice through metagenomic shotgun sequencing. And then we mapped the reads to public databases of known sequences for taxonomy profiling, which means we want to know which bacterial species produce each sequencing read. So this sounds all straightforward, but when we started doing this, we were faced with a bioinformatics challenge. This is because many sequencing reads cannot be mapped to any known sequences in the database. This is due to multiple factors. The sequencing reads are short. On our platform, they are 150 base pairs in length. Also, there is incomplete representation in the database about known bacteria. Also, we are sequencing not from a single genome. We are sequencing from a mixture of genomes, maybe thousands of different bacterial species. Because of this, we found that, especially for control samples, when the diversity is high, up to 90% of the reads cannot be mapped to any known sequences. So in a traditional study, you have to throw away all these reads, all this data. So we decided to try another way. We implemented a novel method to assemble short reads into longer genomic scaffolds. So instead of having 150 base pair short reads, now you can assemble them into longer genomic fragments, up to thousands of base pairs. Now you do mapping, and we found that the mappable reads increased from 10% before assembly to 80% after assembly for control samples, for example. That means now we only need to store away 20% of the data, not 90%. So what did we find from this new approach? Shown here are top 10 bacterial species identified through this assembly approach. Left column is control. Right column is ampicillin treated samples. As you can see, some bacterial species are marked in red. That's because these are known opportunistic pathogens linked to nosocomial infections. For control column, none of the top 10 species are linked to pathogens, but on the right, for ampicillin treated samples, most of the top 10 bacterial species are linked to nosocomial infections. I'm not going to go through every species, but I would like to point out several of them. For example, Acinetobacter baumannii, it emerged in military treatment facilities during the Iraq War, and hence it got a nickname Iraqibacter. Now becomes highly prevalent in civilian hospitals, and some people speculated that this is because of the transfer of patients from military to civilian hospitals. But anyway, now it's common. It can be found in every hospital. It's responsible for 19% of ventilator-associated pneumonia cases, so clearly a notorious nosocomial pathogen. So now let's take a look at another antibiotic, ciprofloxacin. Again, left column is control, top 10 species. Right column is top 10 species from ciprofloxacin treatment. As you can see, many of them are red too. For example, C. difficile, it may colonize human gut without symptoms, but it may expand after the use of antibiotics like clindamycin. And like I said, this is the only pathogen that has a direct link to a specific antibiotic before our study. Is responsible for 200K hospitalizations and 12K deaths in 2017. And another pathogen that's worth mentioning is chlamydia. It primarily causes eye, lung, or genital infections, but can potentially establish long-term infection in gut. And we found that after ciprofloxacin treatment, it's elevated. So the last antibiotic we tested is fosfomycin. Again, 2 out of top 10 pathogens. For example, Bacteroides fragilis is commonly colonizing human gut without symptoms, but it is involved in 90% of anaerobic peritoneal infections. Also can cause bacteremia. It's increased after fosfomycin treatment, but none from control. If we put them together and if we do a statistical comparison, we can see that in this plot, the x-axis are different treatment groups, control or 3 antibiotics. The y-axis are number of reads normalized per million reads. If you look at panel A, that's about the pathogen Acinetobacter, Calcoaceticus, and Bombardier complex. You can see after ampicillin treatment, there's a huge increase of the total number of reads. The reads are color-coded. The orange part are the reads associated with ARGs or antibiotic resistance genes. The blue part are reads not associated with ARGs, but we can focus on the total number of reads. The total length height of the bar is highly increased after ampicillin treatment. If we go to panel G, Chlamydia trachomatis, after ciprofloxacin treatment, the total number of reads are increased a lot, and also some for ampicillin treatment. If you go to panel D, for Bacteroides fragilis, after fosfomycin treatment, the total number of reads increased a lot too. So in a nutshell, we tested 3 antibiotics, and each one of them will cause a huge increase of pathogen reads. So in summary, we found that assembling sequencing reads into longer genomic scaffolds greatly increases the number of usable reads in the metagenome study. And we provided direct evidence for emergence of gut opportunistic pathogens associated with nosocomial infection after the use of all 3 antibiotics, ampicillin, ciprofloxacin, and fosfomycin. Remember, we only tested these 3. If we tested more, we may find more. Of course, whether or not this represents a general mechanism for the emergence of nosocomial infection needs verification from clinical data, but as far as we know, we are the first one to report such direct evidence from in vivo studies for a number of antibiotics. That's all for my talk. I would like to thank the bioinformatics team and also the animal model team from my division, DARS, and also my division leadership, especially Deputy Director Ronnie Rouse, to give support to this project. And that's all. Thanks for your attention.
Thank you, Dr. Lee, for that great talk and discussing the importance of antibiotic treatment and the development of these pathogens. Our speaker will be Dr. Paul Carlson from CBER, who will talk about safety and effectiveness of fecal microbiota transplantation products. Paul.
Thank you. Thanks to the organizers for the invite and to everyone for watching these talks today. I'm just going to jump right in, as always, starting with the typical disclaimer. And then we'll get into the actual meat of the talk here. So a lot of my colleagues have talked about the excitement surrounding the microbiome and the potential for manipulating the microbiome already today. And you've heard multiple people mention C. diff. So these are just some older publications that I pulled for talks in the past. But C. diff has been mentioned, and here's one: feces-filled pill will stop gut infection, specifically referring to using fecal transplants to treat C. diff. And it goes on, and I'll mention some other indications that people are interested in, but here's another one using potential for the gut microbiome to fight obesity. And then one of my favorites here, are you ready to swallow a pill full of poop? And it may be hard to see down at the bottom here, but at the bottom here, it talks about how fecal transplants may be a miracle cure for some of our nastiest illnesses. And as I'm going to go over in the next couple slides, this is the kind of thing that really concerns me in the context of fecal transplants and the microbiome excitement. This graph, I just pulled, I went to PubMed and I searched the term microbiome and then pulled the result numbers by year. And you can see that we have this explosion in microbiome-based studies just in the last 10 years, or 10 or 20 years, 10 to 15 maybe. And this really is leading to a situation where in many instances the hype is getting ahead of the science. Here are a few other websites I recently pulled off, and this demonstrates my concerns and the potential risks here. Do-it-yourself fecal transplant. Let's do this at home. Here's another DIY instructions. DIY instructions, at least this one mentions risks, not just rewards, it mentions both. And here's an article talking about how people are going to YouTube and Facebook for this information. And I just want to pull a couple, point to a couple quotes or a couple pieces of information from these. This one, if you needed a visual, this is going to be one of the best ones of the day for you. This is an MD talking in an interview saying if you've ever made a milkshake, you can do it. Really emphasizing to the general public how easy this is and not emphasizing the potential risks, which are potentially very significant. Here's another, this is from this article about, again, YouTube, Facebook, Reddit. This is where people are going to find their donors. This one was nice that they had a bullet point here specifically saying do-it-yourself FMTs are not recommended by medical doctors. This is very true. There are potential severe risks of doing this yourself. I do want to point out this one comment at the top here. It says the procedure is only FDA approved in a medical setting to treat Clostridium difficile. That is not true. FMT is not FDA approved for any use or indication. I hear that a lot, and that's why I like to mention that when I give talks. FMT is allowed to be used under enforcement discretion for the treatment of C. difficile infections not responsive to standard of care. However, it is not, at this time, an FDA approved drug. It is still considered an experimental drug. All right. So just going to walk through the process really quick, and then I'll get into a little bit of data towards the end here of doing a fecal transplant for those of you who don't think about this every day like we do. We start out with a donor. The stool has to come from somewhere. That donor needs to be screened. So we need to know what to test these individuals for and how good our tests are for doing this screening. And what to test for is going to change over time, as I'm going to talk about in a minute. After we test the donor and that we confirm that the stool is okay, the stool needs to be processed. We have an arm of our research project looking at how that manufacturing process might alter the product, the viability of the organisms, and therefore the efficacy of the organisms. I won't have time to talk about that today, but we're really interested in understanding what happens when these things get exposed to oxygen or some of these other things that are commonly done in the manufacturing process. Once the stool is processed, you have a product. That product needs to be characterized. We need to have a potency assay. How do you measure the potency of this product? It can be complicated because these are complex and highly variable products. How do you measure the stability? How long can it be stored? And a big one there, how do you measure manufacturing consistency with such highly variable starting material? One of the questions that comes up frequently is, does route of administration matter? And I guess the answer is, in some instances, we know that it might, and in other instances, we know that it does not, but we don't really have the answer here. But there are two routes of getting into the gastrointestinal tract, and people are using fecal transplant products going in both ways. We have plenty of capsule-based products or products given by feeding tube, as well as colonoscopy and enema-based products. There's a lot of interest in using fecal transplant. There are a lot of clinical trials underway under IND with the FDA. These are some of the things that have been associated with a dysbiotic microbiome that people are interested in. And this is by no means an all-inclusive list. This is a short list. There are many, many other things that have been added or that could be added to this list. But if something has been associated with the microbiome, then people are interested in studying fecal transplants to treat that. All right, so I want to circle back now for the last half of the talk here to talk about our efforts in testing and in understanding both what we should test for and how good our pathogen tests are. I published this paper in Cell Host and Microbe just over a year ago now, February of 2020. It's nicely outdated already, and I'll tell you why that is. In part, you can probably guess based on what's happened since then. But this gives a good overview of the regulatory considerations for this product class, including a list of commonly performed donor screening tests that I won't have time to go through today. However, I'm going to go over a few safety alerts that have happened in the last few years, many in the last year, and these have been associated with changes in donor screening. So the first of these, we had two instances of transfer of ESBL producing E. coli strains from a donor to a recipient through fecal transplant. One of these two individuals actually died. This changed our donor screening requirements to now requiring not just one or the other, but both of these two things. So donors are now required to both screen for MDROs and to exclude individuals at high risk for colonization from their studies. More recently, we've had cases of enteropathogenic E. coli and Shiga toxin-producing E. coli transfer, and this has also changed our requirements. EPEC is now required. It was not before. And STEC has to be performed by PCR nucleic acid-based testing. It was missed by other methods. And finally, we come to the SARS-CoV-2 and the COVID-19 pandemic, and the fact that this particular virus has shown up in stool. And so we released a safety alert stating that stool donated after December 1st of 2019 should not be used in FMT products until appropriate screening plans can be put in place. I apologize for the noise. My neighbor's cutting his grass. All right, so very quickly in the last couple minutes, I'm going to go through what we have done in this process. We had a question: how can we minimize the risk of SARS-CoV-2 transmission in these products? We know the virus is in stool. The problem was there were no commercially available tests out there for detecting RNA in stool. So we sought to adapt or develop an assay to detect viral RNA in stool that could be used for this purpose. And we determined all of these things, which I'm going to go through briefly. All right, so very quickly, this is our assay. We started out with stool. We got stool that was donated prior to the pandemic from 2 different sources, 5 different donors. We got virus from a colleague at CBER, Tony Wang, inactivated virus that we could spike into the stool, and we did that. And then we went ahead, isolated the RNA, and performed the RT-PCR. We started out with the CDC EUA test because that was the first one that was available when we were working on these things initially in the pandemic. So here you can see our results. After much optimization and effort, we were able to develop a test that could detect virus down to 3,000 copies per gram in stool. This is comparable to what we see with other molecular tests for gastrointestinal pathogens, and it also came in at 600 copies per mil of the solution, the mixture that we made up. But I think the copies per gram value is the more accurate representation here. We did test storage conditions. I'll mention the freezing and I'll skip over some of the others, but we saw that if samples were frozen in various buffers, in PBS or Kerry Blair media, we did see a reduction in the signal. However, if they were frozen in star buffer or DNA RNA shield, we did not see any change in the stability of the RNA in those conditions. And we also went on to test this at 4 degrees and at room temperature, and you can see here some solutions are better than others depending on the conditions that you're using. Importantly, we were able to test this in patients. This was performed in pediatric patients because those are the ones that we could get enrolled at the time. And you can see right here that we were able to detect virus in stool of these individuals in about half of them, showing that this assay works for detection, but maybe not as a strong diagnostic test. And you can get more information on these studies in this paper that we published in Lancet Microbe recently. So in conclusion, we developed an assay that works fairly well. It has multiple potential uses. I've focused on FMT donor screening here today. We showed compatibility with storage. I didn't talk about the cross-lab functionality, but this was very important. We sent samples to another lab, and they were able to implement the entire assay on their own with just our samples. And we showed that this works in pediatric patients. And with that, I'll just acknowledge the people who did the work, in particular Mike Coriel, a postdoc in the lab, who handled the majority of the SARS-CoV-2 assay development. And with that, I will conclude. Thanks for your attention.
Thank you, Paul, for talking about the risk of these FMT products. Our next speaker will be from CISPAN, Dr. Kristen Extra, who will talk about microphysiological systems, regulatory research considerations, evaluation of a model system.
Thank you for the introduction. I'm very excited today to talk about our perspectives for regulatory research considerations with the use of microphysiological systems, using our evaluation of the 2-cell liver chip as an example. So microphysiological systems, or organ-on-a-chip platforms, are miniaturized physiological environments that are engineered to model organ-level responses. They do this with the flow of media over cells, stretch, and multiple cell types. And depending on these design parameters, chips can be used to study a wide variety of tissues, organs, and disease states. Additionally, connecting multiple organ chips together can be used to study multi-organ effects. So due to the growing use of microphysiological systems and a need for new alternative methods, the FDA Foods Program began a creative with Emulate in 2017 to begin addressing questions for the fit for use of organ chips in a regulatory setting. Our laboratory was the first CISPAN laboratory to utilize the Emulate liver chips to answer these questions, and we received funding from DARPA to carry out this research. So we decided to begin with the liver chip because the liver is the primary location of drug metabolism. Drug-induced liver injury is a leading cause of acute liver failure, and liver toxicity is a leading cause of toxicity failures in the pharmaceutical industry. Additionally, the human liver chip has been shown to predict human toxicity that was not predicted by other animal models. So the liver consists of 4 main cell types, including the hepatocytes, which are the most abundant cell type in the liver, and they perform many of the metabolic functions of the liver. The liver sinusoidal endothelial cells, or LSECs, form the lining of blood vessels in the liver. The stellate cells are the fat-storing cells of the liver, and they're important in fibrosis. And finally, the Kupffer cells are the resident liver macrophages, or immune cells. And depending on how many of these cells are included in the chip, you can model the liver in many ways. So we decided to begin with the co-culture containing only the hepatocytes and LSECs, which will be discussed here. But we're also currently working on the more complex quad cell liver chip containing all 4 of those cell types. So here is Emulate's 2-cell liver chip. This PDMS chip consists of 2 channels that overlap, with the primary human hepatocytes seated into the top channel and the LSECs seated into the bottom channel in such a way that they're in close proximity to the hepatocytes. So the remaining components of the Emulate system are involved in the flow of media through the chip. The chips are they're connected to the pods, which contain reservoirs for both fresh and spent media for each of the 2 channels. Sorry, these are the pods. The pods are then placed into these blue bays and loaded into the Zoe, and the Zoe is what is responsible for controlling the rate of media flow through the chip. And then connected to the Zoe is the orb, which supplies the power and CO2 necessary for the Zoe to regulate the flow rate. So in order to evaluate this system, we had 2 main objectives. The first objective was to evaluate the performance of the platform in a regulatory setting, and the second was to look at the quality of the data using compounds of known hepatotoxic potential. So looking at the performance of the system, we found that the Emulate system was easy to use, though there was a little bit of a training period involved. We saw that the critical components of the setup were consistently functional, such as flow rate and critical periods of experimental setup. And we were also able to develop standard operating procedures that were in line with regulatory accepted GLP animal studies. which includes documentation of all the critical processes and ensures data quality. In total, we developed about 40 individual SOPs with about 20 document recording forms. Of particular importance is our chemical evaluation, and this includes testing the solubility and stability of the compounds, confirmation of the dosing solutions, as well as a test material interaction analysis with the CHIPS PDMS. To examine the quality of the data, we looked at its predictivity, sensitivity, and specificity, power, and variability using 8 compounds that were either known hepatotoxic compounds or non-hepatotoxic compounds at 4 concentrations based on their own hepatotoxic profile in order to elicit a wide range of hepatotoxic results. So in order to do this, we set up our experiments, which took about 2 weeks. During the first week, the chips that were set up, which includes activation of the chips, feeding of the cells, as well as preparation of the pods and connection of the chips and pods together. And then during the second week, the chips were exposed to the various test materials for 3 consecutive days. The test material exposure began on day 0, which is exactly 1 week after the hepatocytes were first seeded into the chips. We collected for several different types of analyses. We collected media from days 1 to 3 for a test material interaction with the chip material. Biochemical endpoints were analyzed from days 1 to 3, and the imaging analysis took place on the final day of experimentation. So going into our results, our test material interaction with the chip material showed that there was a wide range of interaction with the chip PDMS, from very high interaction with tamoxifen, and that 100% of the tamoxifen that we loaded into the chip was absorbed by the chip. Alternatively, we had very low interaction with the chip with PDMS, and that only 4% of this compound was absorbed by the chip. So with our biochemical and imaging endpoints, I'm not going to show any specific data because there's quite a bit of it, but instead talk about how useful these assays were for predicting toxicity, as well as what kind of considerations should be taken into account for their use. So albumin was our most predictive assay, and it was our best assay. Urea was a little bit less sensitive and less specific, and lactate dehydrogenase, or LDH, was predictive. However, due to the nature of this assay, consideration needs to be taken into account for the timing to make sure that it coincides with the lysis event. Similarly, with our imaging endpoints, we looked at nuc-blue, which marks nuclei, and this assay was a little bit less predictive in terms of toxicity, but it is a good indication of approximate cell number within the chip, which is a useful tool to have. TMRM, which marks mitochondrial membrane potential, was very predictive, quantifiable, and reliable. And caspase 3-7, which marks apoptosis, was similar to the LDH assay in that consideration of the timing needs to be taken into account to make sure that it coincides with when the apoptosis is happening. So I don't have a lot of time to go over the details of how we did our image acquisition and quantification, but we did recently publish a manuscript that goes into those details more thoroughly. But very briefly, we were able to capture images across the entire length of the chip, as well as through all the layers of the chip. So taking all of our results from the biochemical and imaging endpoints together, we determined that the liver chips accurately predicted toxicity for all of our compounds, except for tamoxifen, which again was shown to interact with the chip material. So after a collection of all of our data, we did a statistical analysis of the system, and here I'm going to compare this to our side-by-side 24-well plate studies, which had hepatocytes only in a dish, and this is the current gold standard for in vitro hepatotoxicity research. So here sensitivity and specificity are shown as area under curve values, and the values that are closer to 1 indicate higher sensitivity and specificity. So here, liver chips had high sensitivity and specificity, so it ranged based on compound and assay, and they generally performed similarly to wells. the notable exceptions are that the liver chips often had higher sensitivity and specificity in the for usnic acid than the well did, whereas for tamoxifen, the well had higher sensitivity and specificity than the chip did. And all these highlighted portions are where one platform did a little bit better than the other. So next we looked at variability both within components of an experiment, such as the Zoe and the Bay and between chips and wells, and between experiments, which is the experiment line. So here's sigma values that are less than 0.2 represent low variability, and as you can see, all of our values are less than 0.2, which indicates consistency of both within and between experiments. So finally, we did a power analysis to determine approximately how many cells or how many chips would be needed to see an effect of a specific size. And here a power of 0.8 or greater is considered acceptable. And for our imaging endpoints, we saw that an N of 3 was needed to see a large effect size, whereas for our imaging endpoints, I'm sorry if I said, I meant biochemical endpoints, an N of 3. And then for our imaging endpoints, an N of 4 was needed for a very large effect size. So to summarize, we saw that the liver chip system was easy to use, though it did require a bit of training. And we were able to develop many SOPs that were in line with GLP regulatory accepted animal studies. And when looking at the quality, we saw that the liver chip system was predictive, except in cases when the chemical interacted with the chip material. And in general, the liver chips had high sensitivity and specificity, high power, and low variability. And we recently published a manuscript that goes into this type of analysis in more detail with one of our compounds, diglycolic acid. And we're also currently working on a manuscript that is going to contain the results from the remaining 7 compounds that is going to look at that type of analysis as well. So to close up, I just wanted to talk about some of the things that we feel are important for consideration in the use of liver chips in regulatory settings. So first of all, we think that it's incredibly important to have good SOPs in place that clearly define all the aspects of the study. And I know I've mentioned this a lot, but most important is the chemical evaluation, especially the chip material interaction with the test materials, because this can have a direct impact on how much of the chemical the cells are seeing within the chip, as well as your interpretation of the results. Next thing to consider is which assays to use and when to use them. Not all of our assays were equally valuable in predicting toxicity, and incorrect timing can lead to missed results. Next, the cost is something to consider. Though liver chips may be a little bit less expensive than a full animal study, they're still quite expensive. The system, the chips, the cells, and all the assays needed to carry out the research all add up, and it's just definitely something to consider when planning these experiments. And finally, though we saw that the liver chips and wells tended to perform similarly on a statistical standpoint, we do think that with increased culture duration and addition of more cell types, more differences may become apparent. And that's exactly where we're moving forward with this. I already mentioned that we're going to be working on the quad cell liver chip, which has hepatocytes, L-sex, stellate cells, and Kupffer cells. And we'll also be looking at longer durations of exposure. So with that, I'd like to thank everyone on the liver chip team, especially Dr. Sprando, who is the PI of this project and planned it out from start to end. I'd also like to thank our FDA management and everyone at Emulate who has helped us out along the way. And that concludes my presentation. Thank you very much for your attention.
Thank you, Kristen, for the exciting talk and giving us the ins and outs of the use of the liver chip. Our next speaker will be from CDRH, Dr. Shelby Scoot, and talk about the evaluation of endothelial cells' responses to nanomaterial using a dynamic flow model. Shelby.
Thank you for the opportunity to present today.
In this presentation, I will give an overview of nanotechnology and medical devices, as well as outline the challenges of biological evaluation of medical products containing nanomaterials and opportunities for the use of advanced in vitro culture models. I will then share data from one of our initial studies using these approaches on the evaluation of endothelial cell responses to nanomaterials using a dynamic flow model. Nanotechnology and medical devices includes a broad range of device applications as well as nanomaterial chemistries and morphologies. Medical products incorporating nanotechnology may include discrete nanomaterials such as metal and metal oxide nanoparticles and or immobilized nanostructures such as nanostructured titanium and electrospun nanofibers. These various types of nanomaterials are being investigated for medical applications such as diagnostics and therapeutics, including drug delivery and implants. Nanomaterials have been shown to influence a variety of biological responses. However, these responses are dependent on the cell and tissue type as well as the nanomaterial physical and chemical properties, such as size, morphology, and charge. Nanomaterial properties are also dependent on the physiological environment, including interactions with proteins and tissues in the dynamic system, making biological evaluation of nanomaterials challenging. Therefore, it is important to assess the nanomaterials in the context of their specific medical device applications. Due to the complex interactions between the nanomaterial physical chemical properties and the biological system, evaluation of biological responses to medical devices containing nanomaterials is difficult, as traditional in vitro assays do not simulate the complex dynamic physiological environment. Additionally, evaluation of nanomaterials using traditional static 2D in vitro culture models can result in assay interferences or nanomaterial cell interactions that do not represent the clinical situation. Due to these challenges, safety assessment of medical products containing nanomaterials relies heavily on animal studies. However, there are ethical considerations, as well as these studies have high costs, are time-consuming, as well as having limitations such as limited disease models and potential lack of predictivity of human clinical responses. Based on these limitations and challenges, there are opportunities to utilize dynamic in vitro culture models to evaluate biological responses to nanomaterials in combination with animal studies for medical device development and safety evaluation. Microphysiological systems such as organ-on-chip technology has shown very promising potential for preliminary drug discovery and toxicity studies by providing more physiologically relevant environment, helping bridge the gaps between traditional in vitro cell studies and in vivo animal evaluation. Therefore, the use of dynamic in vitro culture models have the potential to simulate complex physiological environment and to provide more clinically relevant biological responses to nanomaterials. Our initial research in this area is focused on the use of an endothelium on a chip fluidic model to evaluate biological responses to nanomaterials under shear flow conditions that are similar to human vasculature, as nanotechnology is being increasingly applied for cardiovascular medical applications, and there is potential for nanomaterials in other tissues to enter the bloodstream. For these studies, we first conducted some preliminary studies to evaluate the impact of silver nanoparticles of varying size and concentrations using static in vitro testing to determine the size and concentration of nanomaterials, as well as the endothelial cell model for the development of our endothelium on a chip. We evaluated citrate-coated silver nanoparticles of 10, 20, and 100 nanometer diameter, as well as 4 human-derived endothelial cells, and evaluated cell viability and oxidative stress. These preliminary screening studies demonstrated that the endothelial cell responses were size and concentration dependent, with increased cytotoxicity and oxidative stress observed after exposure to the 10 nanometer silver nanoparticles. Furthermore, the study showed that the human cerebral microvascular endothelial cells, or HCMECs, were the most sensitive to the silver nanoparticles. Based on these preliminary studies, we decided to develop our endothelium on a chip using the HCMECs to evaluate the impact of exposure to 10 nanometer silver nanoparticles. For this study, we first characterized the silver nanoparticles using transmission electron microscopy to confirm the nanoparticle diameter of approximately 10 nanometers. Then, to assess the impact of the biological test system on the nanomaterial properties, we utilized dynamic light scattering to evaluate the hydrodynamic diameter and zeta potential of the in PBS and cell culture medium under static and dynamic flow conditions. These results showed increased nanoparticle size in cell culture medium due to interactions with proteins, as well as increased nanoparticle size under static conditions compared to dynamic flow conditions, potentially due to nanoparticle aggregation under these conditions. These changes to the nanoparticle properties are important to consider in the overall biological evaluation, as they can impact the cell interactions and resulting biological responses. To develop the endothelium on a chip model, we utilized a commercially available dynamic fluidic system to apply unidirectional flow with controlled physiologically relevant shear stress of 10 dyne per centimeter square. For our experimental design, first the human microvascular endothelial cells were seated in the flow chamber and incubated for 72 hours under static conditions to develop a confluent monolayer. They were then exposed to the silver nanoparticles at concentrations ranging from 0 to 40 micrograms per mil under the 10 dyne per centimeter square flow conditions or static control conditions for 24 hours. After the 24-hour silver nanoparticle exposure, endothelial cells in supernatant were collected for the biological assays. The endothelial cells exhibited significantly higher cytotoxicity under flow conditions compared to the static conditions. We also observed increased accumulation of the nanoparticles under the dynamic flow conditions, which was not observed in control studies evaluating silver nanoparticle interactions with the channels in the absence of the endothelial cells. Elevated lactate dehydrogenase or LDH levels were observed in endothelial cells under flow conditions after treatment with 20 micrograms per mil of silver nanoparticles. Similarly, nitric oxide levels were also substantially increased after silver nanoparticle treatment under flow conditions. Caspase-1, an inflammatory response initiator, and Endocan, an endothelial-specific inflammatory response marker, increased after treatment with 20 micrograms per mil silver nanoparticles, suggesting a possible inflammatory response-related toxicity. We further evaluated the endothelial expression of pro-inflammatory cytokines after exposure to silver nanoparticles under static and dynamic flow conditions using a multiplex assay. HCMECs exposed to silver nanoparticles under flow conditions exhibited increased pro-inflammatory cytokines compared to cells treated under static conditions, including IL-1 beta, IL-6, IL-8, and TNF-alpha. Our results show that the dynamic flow conditions affect the silver nanoparticle physical-chemical properties as well as the nanoparticle endothelial cell interactions. We observed differences in the nanoparticle aggregation between static and dynamic culture, as well as observed concentration and flow-dependent biological responses with increased cytotoxicity and increased inflammation compared to cells treated under static conditions. These studies have highlighted the impact of the dynamic physiological environment on nanomaterial physical and chemical properties, as well as the nanomaterial cell interactions and subsequent responses. However, further investigation is needed to determine the underlying mechanisms for these responses. To further develop the model and to expand our research into use of additional cells and organ models, as well as multi-organ models, including commercially available systems, as well as investigation of additional nanomaterials used in medical devices. Nanotechnology is expected to continue to impact the design, development, and manufacture of novel medical products. The performance and safety of medical devices that incorporate nanomaterials depends on the physical chemical properties of the nanomaterials as well as the biological system. Advanced in vitro alternatives have potential to better simulate the complex physiological environment compared to traditional in vitro culture models and to provide more clinically relevant biological responses to nanomaterials. However, currently there are significant limitations on the use of such in vitro models, including uncertainty on translation to in vivo responses. Significant research is needed to validate any advanced in vitro alternative models before broad acceptance in the scientific community and regulatory acceptance for their specific context of use. Although further investigation is needed, there may be advantages for the use of these advanced culture models for product development and biological evaluation of novel technologies used in medical devices such as nanotechnology. I want to thank the many collaborators on this project, particularly postdoctoral fellow Dr. Yizhang Liu, who will be presenting a poster on this project during the FDA Science Forum, And additionally, we would like to thank our funding sources. Thank you.
Thank you. And our last speaker for this section is Dr. Kim Kyung Sung from CBER. And he will be talking about microphysiological systems to assess the functional capacity of regenerative medicine cellular products.
All right, thank you so much for the introduction. So the regenerative medicine is the process of replacing or regenerating human cells, tissues, or organs to restore or establish their normal functions. And the products are typically autologous or allogenic cells that are isolated from tissue biopsies. And it also includes tissue engineered products that are combined with biomaterials and scaffolds. And the field of cell therapy is really rapidly growing, as you can see from this chart. And the number of INDs with the cell therapy development program is rapidly increasing. And we also start receiving several license applications as well. And this is only with cell therapy projects. And our division receives a large number of gene therapy INDs and license applications as well. As you can expect, the regenerative medicine cellular products are very promising and have potential to meet unmet medical needs. But there are still several manufacturing and regulatory challenges that we may need to overcome. The cells are very heterogeneous, and there is patient-to-patient variability, and the source material has limited shelf life and limited sample volume, and there is also limited starting materials for any test method development. And more importantly, our sponsors use a wide range of manufacturing protocols when manufacturing these products. So, and cells are also exposed to different materials and environments during manufacturing. So it is highly possible that the manufactured cells are not identical to the cells freshly isolated from tissue biopsies. So there are critical needs to develop new methods and identify quality attributes that we could use to reliably predict the biological functions of manufactured products. And that is one of the objectives of my research program. And various bioengineering systems such as bioreactors, biomaterials, and microphysiological systems have potential to improve both manufacturing and characterization of cell therapy products as shown in this illustration. And bioreactors and biomaterials may provide better control over chemical and physical microenvironment to enhance the expansion of cell products and maintain their functional properties during manufacturing. And advanced in vitro systems like microphysiological systems can be useful to characterize cell products in a physiologically relevant condition and also identify therapeutically effective cell batches. So in my lab in CBER, we use various microphysiological systems such as organoids and microfluidic systems to develop and improve test methods for cell product characterization and also to identify product attributes that are predictive of safety and effectiveness. And I'm going to present two specific examples. So in this first example, we measured chondrogenic capacity of multipotent stroma cells using the cell aggregate shown in this top left corner. And we study a donor and passage dependency. So we tested 8 different cell lines derived from different donors. And then we used 2 passages that are passage 2 and 3 for early passage and passage 5 for late passage. So we generated this multipotent stroma cell MSC aggregate and cultured them for 21 days in chondrogenic condition, which was created by adding TGF-beta3. And after the culture, we measured 14 shape and size features. And the first 4 images on the left are the representative images of aggregate of early passage MSCs at day 7 and day 21. And the bottom images are from late passage MSCs. And you can see that MSCs at different passages show the different growth pattern, where the cell aggregates from early passages showed some size increase compared to the late passage ones. And to look at the trend, we conducted principal component analysis and generated the score plot shown in the center. And we noticed that the factors associated with the principal component 1 were related to the size feature of the aggregate, and principal component 2 were related to the shape of the aggregate. So the 2 plots shown in the middle shows the principal component 1 value, which is basically a size feature of the aggregate. So you can see that all of the aggregates from the early and late passages showed initial size decrease from day 0 to day 4. And most of early passages MSCs, except the 2 cell lines shown in blue and green, showed size recovery after the first decrease. However, the late passage ones did not show any size recovery except the 1 cell line shown in yellow. And then we conducted histology to detect the deposition of cartilage-related extracellular matrix, such as sulfate GAG shown in blue and collagen shown in red. So as you can see that the cells that showed size recovery deposited higher amounts of extracellular matrix compared to the ones that did not show size recovery. We then looked at the correlation of size feature with the synthetic activity such as GAG and collagen production and chondroid gene expression. And it was very interesting to see that the synthetic activity, but not chondroid gene expression, correlated with the size features by day 7. So the take-home message from this study is that our high-throughput and non-destructive approach, which basically measures the size of the aggregate, could potentially serve as a tool to identify MSC lines that are more chondrogenic by day 7 instead of day 21. So therefore, this could really improve the manufacturing strategies for MSC-based cellular products for cartilage tissue repair. And in the first example I just presented, we looked at the direct differentiation of MSCs into cartilage-forming cells. But MSCs are also known to produce a lot of paracrine factors that could be beneficial for wound repair, immunomodulation, ischemic reperfusion. So in this second example, we study whether we can develop a new quantitative assay to measure the paracrine potential of MSCs to induce vasculogenesis. So in this second example, we use the compartmentalized co-culture platform to co-culture MSCs with endothelial cells to look at the MSC's vasculogenic potential. And like the chondrogenesis project, we look at both donor and passage dependency, as you can see from the configuration table. And we use lung fibroblasts as a positive control, since it is well known that the lung fibroblasts induce vasculogenesis. And the images shown in the bottom left are the representative images of endothelial cells co-cultured with different MSC lines and lung fibroblasts. And as you can see from the image, the extent of vasculogenesis was different between MSC lines. And since we are interested in developing quantitative assay, we are currently quantifying the extent of vasculogenesis using different algorithms. And as an example, on the right side, we have a bar graph showing average vessel length and the number of junctions. And then the first 2 bar graphs on the top shows the donor dependency. So the first 4 graphs, blue, green, orange, red, are from different MSC lines, and we have lung fibroblasts in pink and endothelial cell monoculture in purple. And the bottom 2 shows the passage dependency, where blue is from passage 3 and green is from passage 5. So even though this is not complete data set, these data suggest that vasculogenic potential of MSCs appear to be influenced by manufacturing parameters, such as the donor source and passage number. So we think that it is important to have a robust and reliable assay to understand the functional heterogeneity of the product and also the influence of the manufacturing parameters. So in my presentation, I showed 2 examples of how we utilize microphysiological system to evaluate functional capacity of cellular products. And I think that the various microphysiological systems could be very useful in improving our understanding of cell-cell interactions, cell materials interactions, and also it may improve our understanding on the impact of the numerous manufacturing parameters. So with that, I'd like to acknowledge my lab members. These are wonderful, hardworking people. And also I'd like to acknowledge my collaborators and funding sources. Thank you very much.
First of all, I'd like to thank all of the speakers in this session 5 today, advancing products based on novel technology. I would like to say that we met our learning objectives. We discussed the potential utility and challenges of these new technologies. So in closing, I would like to just go over a few of the advantages of these systems. So in evaluating these in vitro microphysiological systems for organs on a chip, there are several advantages. These systems can represent major organ tissues in the human body, which can facilitate the assessment of biomarkers, bioavailability, efficacy, and toxicity of therapeutic agents or preclinical studies. Also, they can provide insight into normal human organ function and disease pathophysiology. They can bridge gaps between traditional in vitro cell studies and in vivo animal evaluations and further provide prediction of clinical responses for safety assessment. These models employ the use of multiple cell types and flow media to mimic physiological stimuli. So advances in development of these models using sophisticated integration of biology and engineering, as you saw in the presentation today, they have resulted in the development of models for human micro interaction, as we examined in these presentations. We further discussed in this section the importance of the microbiome in relation to the microphysiological system. The microbiome is a key contributor in the metabolism of drugs, food additives, pesticides, herbicides, and other contaminants. There are knowledge gaps between the interaction of these systems with the host and the microbiome of these microphysiological systems. So therefore, increased understanding of the impact of antimicrobial drug residues on the human intestinal microbiome is needed, including the development of antimicrobial resistance, as discussed in some of our presentations today. These systems could improve the mimicking of human intestinal physiology and microbial complexity to address these gaps, as also shown in a number of the presentations today. However, there are challenges, as was discussed by a number of the scientists in their presentation today. One of the major challenges as we go forward with the system is the acceptance of these models as new tools for use in regulatory settings. Furthermore, the validation and the qualification with these models, will they produce results that are consistent? Also for the need for the development of standards for regulatory acceptance of these novel products, as we've seen. So, the question that I will leave you with today, as we close out these sections, are these the tools for regulatory decisions in the future? Validation of these models are currently being conducted not only as we discussed today, but throughout the country in a number of laboratories. Also in the various centers. We had representatives from each of the FDA centers discussing these models of technology today. Therefore, to better understand the usefulness of this technology. So, as I close out this section today, we did not have an opportunity for questions for our speakers, but I would like to further thank Dr. Linda Griffin, who kicked off this section Forrest is our keynote speaker from MIT. I think this was a very informative section, and therefore we will now end this section. But before we end this section, take our lunch break, we would like to encourage you. There are a number of posters in this section that will further discuss other models or other systems that was not discussed in our presentation. So please go and visit those posters sections. Also, before, if you have any questions, please, you can contact me. I am Beverly Lane Cook, who has served as your moderator for today. And my contact information is I am at the National Center for Toxicological Research. So as we continue to discuss and close this section today, I would like again to thank each of the speakers for their presentation. As I said again, the use of these models are great potential for regulatory science. At this time, I will like to further discuss the interaction of these models with the microbiome. And also, one of the presentations that was highlighted in our talk today, and it really focused on the fecal transplant and the use of these potential products without the approval of the FDA. And also, because of the COVID infection, I thought that presentation really brought home the use that these models should be evaluated very closely for use for potential diseases. They have a lot of potential use to be used. However, but consideration like the media, Facebook, and all of the information that sometimes is given to the public, it is important for individuals to really evaluate what is being shown in these media platforms. So again, I would like to say that advances in bioengineering and material science, as stated in our first talk by Dr. Griffin, is critical. And also it is critical for the development and the understanding of the various platforms that are being utilized in these microphysiological systems. So today we have discussed in the series of these talks the possibility of the integration of these novel alternative models into the potential regulatory environment that can be used to address a number of unmet public health concerns, better models of human adverse responses, as well as identifying and evaluating more reliable biomarkers for monitoring toxicity, as well as the interaction with the microbiome. As I said before, there are a number of challenges of these new systems, as well as understanding them with advancing these product development. So, as I close this section out, again, I would like to thank all our speakers today that represented all of the centers. We were able to get this section and make sure that all of the scientists from all of the centers was represented to discuss these new technologies for advancing our products. Again, I would like to thank our speakers today. Also, it is important we want our speakers for the responses for nanomaterials and use of these systems. We try to address every aspect that could be in addressing these new technologies. So I think we have, as I said before, met our objective for this section. We discussed in detail the importance of these systems as well as the challenges of these systems and the importance of integrating this knowledge into scientific communication with regulatory and research work. Again, as we close, The question I'll leave with you today is, are these tools for regulatory decisions in the future? Are these the tools? And therefore, as we continue to validate these modes, let's think about that, and also think about in the future, are these alternative models capable of replacing or assisting animal model studies for regulatory decisions as we journey through these sections. As we close, I would like to again encourage you, as we take this lunch break until 12 o'clock, there will be 2 concurrent sections in the afternoon. Please visit the poster exhibit. Again, thank you, and I hope you enjoyed section 5.